Welcome to the deep dive. Have you ever looked at a plant, maybe a towering tree or just a tiny sprout, and wondered how it's actually built? How does it stand up? How does it move things around inside? Today, we are taking a deep dive into the fundamental architecture of plants, drawing key insights from a foundational chapter in Plant Physiology and Development, 6th edition. We're going to use this source as our guide, moving from the plant's overall structure, its tissues and organs, right down to the incredibly complex world happening inside individual plant cells.
It really is like getting the ultimate blueprint. And it's much more than just listing parts. This chapter helps us understand how these different components are organized, how they work together dynamically, how they develop over time, and the amazing processes that let plants live, grow, and, well, interact with their environment without being able to just get up and move.
Exactly. So our mission today is to navigate this chapter, pull out the most important nuggets of knowledge about plant and cell architecture. Understand the essential building blocks and see why knowing this stuff matters. Why does the structure of a cell wall affect how a plant grows? How do cells communicate when they're locked in these, rigid boxes? Let's unpack this and find the core insights. Okay, let's start broad. Plants seem incredibly varied. From the smallest flirting speck to the largest living things on Earth. How could they possibly share a basic architectural plan?
It is remarkable, isn't it? The chapter points out that despite this huge diversity in form and size, all plants share fundamental principles. Think about it. They all capture solar energy through photosynthesis, right? They grow to find resources, light, water, nutrients, because, well, they can't move to get them. They've evolved complex vascular systems to transport water, minerals, and sugars all through their body. They have rigid structures, largely defined by their cell walls, which is just crucial for standing upright. And they've adapted amazing ways to survive life on land, preventing things like drying out. These are shared, defining characteristics.
And their life cycle is fundamentally different from animals, too. There's this whole concept of alternation of generations.
Right. That's a core part of the plant blueprint. Plants cycle between two distinct multicellular bodies. One is diploid, two sets of chromosomes, the sporophyte generation. The other is haploid, with just one set of chromosomes. That's the gamephyte generation. These two generations take turns in the life cycle. How prominent each one is varies a lot depending on the plant type, but the basic principle is shared across the board.
So, putting aside the life cycle for a second and the huge visual variety, there's a common underlying structural organization.
Yes, absolutely. The source really emphasizes this basic body plan built upon three major tissue systems found in all plant organs, whether it's a root, a stem, or a leaf. You have the dermal tissue, that's the outermost layer, kind of like the plant's protective skin. Inside that is the ground tissue, which makes up the bulk of the plant body, doing jobs like storage, support, photosynthesis. And weaving through it all is the vascular tissue, the xylem and phloem, forming the transport network, basically the plant's circulatory system.
Okay, that makes sense at the organ level. But zooming in on the cells themselves, there's a key difference in how plants build their bodies compared to animals. Plant cells don't migrate, right?
Exactly. And this is a crucial insight from the chapter. Because plant cells are encased in a rigid cell wall, their development depends entirely on the pattern of cell division, where and in what directions cells divide, and then the subsequent enlargement of those cells. They can't just crawl into place like many animal cells do.
That cell wall seems pretty fundamental then.
Okay, so because they're in these rigid boxes, how do cells even talk to each other or share resources?
This leads us to one of the coolest features. Plasmodesmata. These are microscopic channels that connect the cytoplasm of one plant cell directly to its neighbor. They pass right through both cell walls and the plasma membranes. Think of them as tiny cytoplasmic bridges or tunnels.
Tunnels connecting cells. That's fascinating. They mention different kinds, like ones formed when cells divide and ones that form later. And there's even a thread of endoplasmic reticulum running through the middle of the tunnel.
Yes, the desmotubule. It's a structure derived from the ER running down the center of the plasmametal channel. Exactly how it functions is still being researched, but it's a direct membrane connection across cell boundaries. The channels also have structures like callus collars that can potentially regulate the size of the opening, controlling what molecules can pass through. This pathway, the symplast via plasmodesmata, is just vital for communication and transport throughout the plant. So plants build structure by dividing cells, which are constrained by cell walls but connected by plasmodesmata. And where does this division happen to make the plant grow?
That's the job of meristems. These are specific regions in the plant body containing actively dividing cells, essentially stem cells for plants. You have apical meristems at the tips of roots and shoots, responsible for primary growth, in length. Axillary buds in the leaf axils are also meristems, waiting to become branches. And then there are lateral meristems, like the vascular cambium, which cause secondary growth, increasing the plant's width.
Right, secondary growth is what makes trees get wider, adding wood.
Precisely. The vascular cambium is a cylinder of actively dividing cells. It produces secondary xylem, which is the wood, towards the inside, and secondary phloem towards the outside. This process adds girth and strength, especially important for tall, woody plants. And the chapter highlights how, in areas with seasons, the difference in growth rate between spring and summer leads to those visible annual rings we see in wood. Tells you about the plant's age and past growing conditions.
Okay, so we have the big picture. Plant body plan, growth from meristems defined by rigid cell walls, connected by plasmodesmata. Now, let's shrink down even further.
Right.
What's actually inside one of these plant cells?
Right. Inside the cell wall and the plasma membrane, plant cells have the familiar eukaryotic components. A nucleus, cytoplasm, various membrane-bound organelles. But they also have some unique features, or particularly prominent ones, like the large central vacuole. The chapter actually categorizes organelles by their origin. Some are part of the endomembrane system network, others function and divide more independently, and some, like mitochondria and plastids, even have their own DNA, which hints at their ancient origins.
Let's start with the outer boundary inside the wall, the plasma membrane. It's more than just a sac holding everything in, I take it.
Oh, much more. It's a dynamic barrier following the fluid mosaic model, a flexible lipid bilayer with proteins embedded within or associated with it. That fluidity is key for its functions, like transport and signaling. And the chapter notes how plants can adjust the fatty acid composition of their membranes. They add more unsaturated fatty acids, which are kinkier, and prevent tight packing to maintain fluidity in cold temperatures. That's a real challenge for plants. And those embedded proteins are the real workhorses, doing everything from moving specific molecules across the membrane to receiving signals from outside the cell.
And the nucleus, the cell's command center, where the genetic information lives.
Right. It houses the nuclear genome, the primary set of instructions. The chapter mentions the fascinating variation in genome size across plant species. Some have incredibly compact genomes, like Arabidopsis, while others have absolutely massive ones. It's quite a range. The nucleus is enclosed by the nuclear envelope, a double membrane punctuated by nuclear pores that control what goes in and out. Very selective. Inside, the DNA is organized with proteins into chromatin. It exists in more open, active forms, that's euchromatin, or more condensed, less active forms, heterochromatin. And the concept of epigenetic regulation, heritable changes in gene expression without changing the DNA sequence itself, is highlighted as really crucial for plant development in response to the environment. And of course, the nucleolus inside is where ribosomes are built.
Next up, the endomembrane system. The cell's internal network for making, modifying, and shipping things.
Yeah.
Sounds busy.
It is a bustling factory and delivery system. It includes the endoplasmic reticulum, or ER, which is this vast dynamic network of interconnected membranes. There's rough ER, studded with ribosomes, where proteins destined for secretion or other organelles are made and folded. And smooth ER, involved in lipid synthesis, detoxification, calcium storage. The ER forms different structures, sheets, tubules, even those transvacuolar strands crossing the central vacuole, and these can rapidly change form, influenced by proteins and the cytoskeleton. The ER also plays a role in establishing the asymmetry of membranes. It's very dynamic.
And the Golgi apparatus, which plants often call a dictyosome. What's its role?
The Golgi is like the cell's processing and packaging plant. It's a stack of flattened membrane sacs called cisternae. It receives proteins and lipids from the ER, further modifies them, sorts them, and packages them into vesicles for transport to their final destinations, whether that's secretion outside the cell, delivery to the vacuole, or incorporation into other membranes. It's particularly important in plants for synthesizing and processing polysaccharides that end up in the cell wall.
Okay, so how does stuff actually move through this Golgi stack? Is it like a conveyor belt?
It's a bit more complex than that. It involves vesicles budding from one cisterna and fusing with the next, but also a process called cisternal progression, where the cisternae themselves mature as they move through the stack. There's also retrograde transport, using different types of vesicles to send enzymes back to earlier compartments. It's about maintaining the right processing environment. And finally, vesicles bud off from the trans-Golgi network, the sorting station at the end, to deliver cargo to the plasma membrane or vacuole. It's a constant flow of membrane traffic, really quite intricate.
Let's talk about the big one, the vacuole. In mature plant cells, it can take up most of the cell volume. It looks like an empty bubble, but you're saying it's definitely not.
Absolutely not empty. The vacuole is incredibly versatile and crucial for plant life. Its huge size in mature cells is key for cell expansion. It pushes the cytoplasm against the cell wall to increase cell volume efficiently. It's a storage depot for ions, sugars, amino acids, waste products, even pigments that give flowers color. And critically, it maintains turgor pressure, that internal hydrostatic pressure against the cell wall. That's what keeps a plant rigid and prevents wilting. Without turgor, plants just go floppy. It also has a lytic function, acting like a plant lysosome to break down unwanted molecules or organelles. And the membrane surrounding it, the tonoplast, has powerful proton pumps that accumulate solutes inside, helping build that turgor pressure. Super important.
Are there other interesting smaller organelles worth mentioning?
Yeah, the chapter mentions things like oil bodies for storing lipids and microbodies such as peroxisomes and glyoxysomes, which are involved in specific metabolic reactions. Like mitochondria and plastids, these can grow and divide independently of the main cell cycle.
And speaking of mitochondria and plastids, these are often called the powerhouses, right?
Exactly. Mitochondria are where respiration happens, generating most of the cell's ATP energy. Plastids are a diverse group, including chloroplasts, the sites of photosynthesis, converting light energy into chemical energy. The crucial insight here, as the chapter points out, is their endosymbiotic origin. They were once independent bacteria engulfed by early eukaryotic cells. This history means they retain their own small circular genomes and divide by fission separately from the cell's nuclear division.
So there's semi-autonomous little entities inside the cell. Fascinating. And plastids can specialize, you mentioned.
Yes, they are quite plastic adaptable. Chloroplasts contain chlorophyll for photosynthesis, obviously, but there are also chromoplasts, packed with pigments that give fruits, flowers, and autumn leaves their vibrant colors. Think reds, oranges, yellows. And leucoplasts, which are non-pigmented and primarily function in storage, like accumulating starch in roots or potatoes.
It sounds like there's a lot of static structure, but you also hinted at a lot of movement inside the cell.
Oh, absolutely. The chapter highlights that organelles like mitochondria and Golgi stacks are constantly moving around the cell. They don't just float passively. They're actively transported along tracks by motor proteins. It's a really dynamic process, quite rapid in some cases.
Which brings us perfectly to those tracks, the cytoskeleton.
Right. The cytoskeleton is the cell's internal framework and railway system, if you like. It's a dynamic network of protein filaments. It provides structural support, helps maintain cell shape, organizes the organelles, and acts as tracks for all that movement we just talked about.
What are the main components in plants?
In plants, the two main types are microtubules, made of the protein tubulin, and microfilaments, made of actin. Both are highly dynamic structures, constantly assembling and disassembling. Cortical microtubules, located just beneath the plasma membrane, are particularly important in plants because they guide where cellulose is deposited in the cell wall, and that ultimately determines the direction the cell expands, which shapes the whole plant. Organelle movement along these tracks is powered by different motor proteins. Myosins work with actin filaments, while kinesins and dyneins work with microtubules. It's worth noting, as the chapter does, that plants lack the intermediate filaments found in animal cells, so the system is a bit different.
So the cytoskeleton is essential for structure, internal transport, and even directing cell wall formation. That's a lot.
And it plays an absolutely critical role in cell division, orchestrating the movement of chromosomes and the formation of the new cell wall.
Speaking of cell division, this is how plants make new cells for growth, right?
Exactly. New cells arise from pre-existing cells through the cell cycle, which includes DNA replication and then the actual division, mitosis, and cytokinesis. This cycle is tightly regulated by a complex network of proteins, notably cyclins and cyclin-dependent kinases, ensuring that cell division happens at the right time and place. It's very controlled.
And the actual division of the nucleus, mitosis itself?
Mitosis is where the duplicated chromosomes are accurately separated into two new nuclei. Plant mitosis involves the condensation of chromosomes, the formation of a spindle apparatus made of microtubules. This spindle captures and aligns the chromosomes at the cell's equator, the metaphase plate. Then proteins like the anaphase promoting complex trigger the separation of sister chromatids, pulling them to opposite poles. Plant spindles differ slightly from animal spindles in how they are organized at the poles, but the basic outcome is the same. Identical sets of chromosomes for each daughter cell.
And after the nucleus divides, the cell itself has to split into two. How does that happen with a rigid cell wall? It can't just pinch like an animal cell.
No, it can't. This is a key difference. Plant cells form a cell plate down the middle. This process involves a structure called the phragmoplast. It's made of microtubules and actin filaments, and it guides vesicles carrying cell wall material to the cell's equator. These vesicles fuse, building the new cell wall and plasma membrane outwards from the center, until they reach the parent cell walls, dividing the cell into two daughter cells. A neat detail the chapter mentions is how some gets trapped within the forming cell plate. This establishes those primary plasmodesmata connections right from the start, linking the new cells. And the orientation of the cytoskeleton just after division also influences the direction of future cell expansion, linking cell division directly to the plant's overall form.
Wow. So from these basic dividing cells, plants generate this huge variety of specialized cells that make up the tissues we talked about earlier: dermal, ground, vascular.
Yes, exactly. Differentiation leads to distinct cell types within those systems, each adapted for specific functions. In the dermal tissue, the epidermis, you find specialized cells, like those puzzle-shaped pavement cells or guard cells that form and regulate stomata, the pores, for gas exchange. Super important for photosynthesis and preventing water loss. Stomatal opening is influenced by things like blue light sensing and changes in guard cell purgar pressure driven by ion movements. It's quite sophisticated. You also find trichomes, or plant hairs. They can provide protection, or, like in the example of Abutilon flower nectaries, be specialized secretory cells producing nectar. The chapter details how these secretory cells often have lots of and plastids involved in producing the substances, and how secretion can involve plasmodesmata and the
Yeah.
space outside the plasma membrane.
Okay, what about the ground tissue, the bulk of the plant? What kinds of cells are in there?
Ground tissue contains versatile parenchyma cells. They do storage, metabolism, lots of things. Then you have collenchyma cells, which provide flexible support in growing tissues thanks to their thickened primary walls. And sclerenchyma cells, providing rigid support with their heavily thickened, often lignified secondary walls. Sclerenchyma includes tough fibers and irregularly shaped sclerids, like the gritty stone cells in pear flesh or the ones that give structure to camellia petals. The chapter even highlights how environmental factors can influence sclera development. And another specialized ground tissue layer is the endodermis in roots. It has a waxy barrier called the Casparian strip, which precisely controls which water and solutes can enter the vascular tissue from the soil. Very important for selectivity.
And finally, the vascular tissues, the transport system. Xylem first.
Right. Xylem conducts water and minerals upwards from the roots. Its main conducting cells, tracheids, and vessel elements are actually dead at maturity. They leave behind hollow tubes with reinforced, often lignified secondary walls containing pits that allow water flow between cells. The cytoskeleton plays a role in guiding where these strengthening walls are deposited during development.
Okay, and phloem transports sugars produced during photosynthesis throughout the plant.
Correct. Phloem-conducting cells are sieve elements in flowering plants. They're called sieve tube elements, connected end-to-end by perforated sieve plates. Unlike xylem, sieve elements are living, but they lack a nucleus and many other organelles at maturity. They are very closely associated with companion cells. These are fully functional cells connected to the sieve elements by extensive plasmodesmata. Companion cells provide metabolic support and are crucial for loading sugars into the sieve elements for transport. It's a real partnership.
Wow. So we've really gone from the entire plant body and its growth strategy down to the dynamic world inside a single cell, its organelles, cytoskeleton division, and then zoom back out to see how these cells differentiate to form specialized tissues with distinct jobs. It's quite a journey.
It really gives you a sense of the integrated architecture, from the shared body plan and growth from meristems, defined by that cell wall and interconnected by plasmodesmata, to the dynamic processes within each cell, the membrane trafficking, the organelle movement, the precise mechanics of cell division, and how all this culminates in specialized tissues performing vital functions like transport, support, and protection. It all fits together.
And understanding these fundamental building blocks is absolutely key, isn't it?
Yeah.
For understanding everything else in plant biology, how plants respond to light, how they defend against pathogens, how they produce fruits or flowers, it all starts with this basic architecture.
Exactly. And maybe a final thought to leave you with. Consider how the rigid cell wall, which might seem like a limitation, has actually been a fundamental driver in plant evolution. It's forced them to develop unique strategies for growth, communication, and adaptation that are completely different from organisms with more flexible cells, like animals. What aspect of this deep dive into plant architecture did you find most surprising or insightful?